In today's video we shall discuss about inhibitors of electron transport chain and uncouplers of oxidative phosphorylation. But before that, do subscribe to my channel and let me know your opinions and suggestions in the comment box. The electron transport chain is a series of chemical reactions that occur in the inner membrane of the mitochondria in eukaryotic cells or the plasma membrane of prokaryotic cells. It is responsible for the production of ATP through oxidative phosphorylation. Inhibitors of the ETC can disrupt the electron flow, which can lead to a decrease in ATP production, oxidative stress, and cell damage. Some of the inhibitors of electron transport chain are rotenone, cyanide, mixothiazole, antimycin A, pyricytin A, and oligomycin. Rotenone. Rotenone is derived from the roots of several plants, including the jicama vine and the derris plant. It is a natural insecticide and a potent inhibitor of complex 1 of the ETC. It binds to the iron-sulfur clusters of complex 1 and prevents the transfer of electrons from NADH to ubiquinone. This leads to a decrease in ATP production and an increase in reactive oxygen species production. In addition to inhibiting complex 1 of the ETC, rotenone has been shown to have neurotoxic effects and has been associated with an increased risk of Parkinson's disease. Cyanide. Cyanide is a highly toxic chemical that can be found in various industrial processes, such as gold mining and metal plating. It is also produced naturally by some plants, such as the cassava plant. Cyanide inhibits complex 4 of the ETC by binding to the heme group of cytochrome C oxidase and preventing the transfer of electrons to oxygen. This leads to a rapid depletion of ATP and can cause cell death within minutes. Mixothiazole. Mixothiazole specifically inhibits complex 3 in the ETC by binding to the ubiquinol oxidation site, which is a key component of the electron transfer pathway. Mixothiazole disrupts the flow of electrons through the ETC, leading to a decrease in the production of ATP and an impairment of cellular respiration. This can have a significant impact on cellular metabolism and function. Additionally, mixothiazole is also known to generate reactive oxygen species, which are highly reactive molecules that can cause cellular damage and contribute to oxidative stress. Antimycin A. It is a fungal antibiotic that binds to complex 3 of the ETC and inhibits the transfer of electrons from ubiquinone to cytochrome C. This leads to a decrease in ATP production and an increase in ROS production. Antimycin A is produced by several species of Streptomyces bacteria and is commonly used as a research tool to study the ETC. Like rotenone, it can be toxic to cells and has been shown to induce oxidative stress and apoptosis. Pyricytin A. Pyricytin A is a natural compound that acts as an inhibitor of complex 1, NADH dehydrogenase. It is produced by several microorganisms, including Streptomyces and Streptococcus species. Pyricytin A inhibits complex 1 by binding to its quinone binding site, which is responsible for the transfer of electrons from NADH to ubiquinone, a crucial step in the ETC. By binding to this site, pyricytin A disrupts the flow of electrons through complex 1, leading to a blockage of electron transfer and subsequent impairment of ATP production. Oligomycin. Oligomycin is produced by Streptomyces diastereochromogenes and is commonly used as an antibiotic. It inhibits ATP synthase, the enzyme responsible for the final step in ATP production. It binds to the F0 subunit of ATP synthase and prevents the flow of protons back into the mitochondrial matrix, which leads to a decrease in ATP production. In addition to these inhibitors, other compounds can also interfere with the function of the ETC. For example, some drugs used in chemotherapy, such as doxorubicin and cisplatin, can cause damage to the ETC and lead to oxidative stress and apoptosis. Atpinin A5 and CO, CCCP are other examples. Oxidative phosphorylation. Oxidative phosphorylation is the process by which the energy released from the oxidation of nutrients is used to produce ATP in mitochondria. Uncouplers of oxidative phosphorylation are compounds that disrupt the coupling of electron transport and ATP synthesis by dissipating the proton gradient across the mitochondrial inner membrane. This results in a decrease in ATP production and an increase in metabolic rate, as the electron transport chain continues to function but is no longer coupled to ATP synthesis. Some of the uncouplers of oxidative phosphorylation are valinomycin, FCCP, DNP, atrocytoside, thermogenin, and CCCP. Valinomycin. Valinomycin is a natural compound produced by some strains of Streptomyces bacteria. It is a potassium ionophore, meaning it can transport potassium ions across the mitochondrial membrane, which can disrupt the proton gradient and uncouple oxidative phosphorylation. 2,4-dinitrophenol. DNP disrupt the coupling between electron transport and ATP synthesis by dissipating the proton gradient across the inner mitochondrial membrane. These compounds increase the permeability of the membrane to protons, allowing protons to leak back into the mitochondrial matrix without passing through ATP synthase. This process is known as proton leak, and it prevents the proton gradient from being maintained and used to drive ATP synthesis. Thermogenin. Thermogenin or uncoupling protein 1 is a physiological uncoupler of oxidative phosphorylation found in brown adipose tissue. UCP1 is a mitochondrial protein that uncouples oxidative phosphorylation from ATP synthesis by facilitating the transport of protons across the mitochondrial inner membrane, which dissipates the proton gradient and generates heat instead of ATP. This process is essential for maintaining body temperature in mammals, especially in newborns and hibernating animals. UCP1 provides an alternative pathway by which protons can re-enter the mitochondrial matrix from the intermembrane space, instead of going through ATP synthase. The result is a temperature increase rather than ATP production in the tissue containing this protein family. FCCP. FCCP stands for carbonyl cyanide paratrifluoromethoxyphenylhydrazone. It is a chemical compound that is commonly used as an uncoupling agent of oxidative phosphorylation in cellular and mitochondrial studies. FCCP is a proton ionophore, which means it facilitates the movement of protons across the mitochondrial membrane, dissipating the proton gradient and uncoupling oxidative phosphorylation from ATP synthesis. This leads to an increase in oxygen consumption and heat production, as the energy generated by electron transport is dissipated as heat instead of being used to produce ATP. Atrocytoside. It is a naturally occurring compound derived from the thistle plant and has been studied for its effects on mitochondrial function. Atrocytoside acts by disrupting the normal coupling between electron transport and ATP synthesis in mitochondria. It specifically inhibits the adenine nucleotide translocase, which is a mitochondrial membrane protein responsible for the exchange of ADP and ATP across the mitochondrial inner membrane. CCCP. CCCP, or carbonyl cyanide M-chlorophenylhydrazone, is a chemical compound that is widely used as an uncoupler of oxidative phosphorylation in research settings. It is a synthetic compound that acts as a protonophore, meaning it allows protons to freely pass across the mitochondrial inner membrane, uncoupling the electron transport chain from ATP synthesis. The diagram here shows the coupling and uncoupling of oxidative phosphorylation. To conclude, inhibitors and uncouplers of the electron transport chain and oxidative phosphorylation are powerful tools for studying cellular respiration and energy metabolism. By selectively blocking or disrupting specific steps in these pathways, these compounds can provide insights into the regulation of ATP synthesis and the generation of cellular energy. Moreover, inhibitors and uncouplers have potential applications in treating various diseases, such as cancer and metabolic disorders, by targeting the energy metabolism of abnormal cells. However, the use of these compounds must be carefully controlled, as they can also have adverse effects on normal cellular processes and cause toxicity.